couple of minutes about FyoniBio, a bit about our company first, and then I brought a case study which we recently went through, which was to make it a bit more interesting for the audience, not only taking or talking about things what we could do, but as well things what we have done and how we could really help our customers. First of all, a bit about FyoniBio. So we are Berlin-based here in the northern part at the campus from the Max Delbrück Center. Many know that, I guess, in this context. We are a young company, about 3.5 years old right now, but we offer 15 years or over 15 years in CDMO expertise. And this is, like Annika just told before, because we are more or less originating out of the company Glycotope. Glycotope was active from 2000 and developed biopharmaceuticals through clinical phases until phase 3 ready. And in the peak times, the Glycotope was here in Berlin as a core research and development unit and had a GMP manufacturing site in Heidelberg. Well, biotech is like biotech is, so there's not a guarantee for having a retirement contract in this business. So after 2 major clinical trials went wrong for Glycotope, then there was a major shrinkage in the company. And in the second wave, Hans Baumeister, my friend and colleague, and myself decided to form a unit which we can spin out to a company which is then offering exclusively services to anybody on the world who's asking for that. So, and we did that on 1st February 2022 and started the business at an Glycotope independent company, which we called then FyoniBio. With that, we seamlessly transferred the ISO certificate to the new company and started 3.5 years ago with the business. This slide shows the areas where we are offering business, and this is basically in the CMC development. So that's where the title of the talk today comes from. But as well, further downstream, we are offering services in clinical settings. So when companies go to phase 1, phase 2, phase 3 clinical trials, we can as well help with clinical bioanalytics. And in this context, if you take a look at the time scale from start of a project to market, we are very strong in the preclinical development part, mainly CMC, like cell line development, process development, but as well in the context of characterizing the molecules with bioassays, cell and non-cell based bioassays. We are offering a very sophisticated mass spec analytics, which we offer independently to any projects or in combination with cell line and process development projects to the customer, and that goes really nicely hand in hand with all the offerings what we are serving, because often when it comes to biopharmaceuticals, it's not only the protein, but the protein is modified by glycans, O and glycans, and these have to be monitored very precisely to not get some surprises when it goes into animals or humans at the end. Clinical bioanalytics can be served then from the first phase to post-market studies. And what we do not offer in-house, but what we offer with reliable partners around the world is the GMP manufacturing. And that can be shown on this slide. So we are currently building up and is still growing a network of GMP manufacturers around the world. This is quite interesting as well for our customers, because they cannot only choose from one bioreactor type, one technological setup, what we might offer ourselves. We decided last year to not offer that ourselves, but to partner with different companies around the world. And you see that kind of from China, India, two sites in Europe, one currently in Northern America, that the region around the world is nicely covered depending on kind of the origin of our customers. They might want to have some local manufacturers as well as from technology, prices, drug substance, drug product manufacturing. So there are many differences between all these GMP manufacturers, and that can be taken into account, and our customer can choose within a wide variety of possibilities. What is kind of the basis? One of our pillars is certainly the cell line development. The other pillar is the clinical laboratory, what we are offering. But because I am more taking care of the CMC part, the talk will be about that area. And here are the platforms what we are offering at FyoniBio. We are only offering mammalian production technologies, so no yeast or E. coli production. But when it comes to mammalian production, I think we are one of, if not the broadest offerer of different platforms around the world. We do have two CHO platforms. One is a classical CHO portfolio. I will come to that on the next slide. Other ones is a food aid knockout cell line after the off-patent situation from the Protelligent technology from the Japanese company Biova. We have as well launched such a technology with up-to-date cell lines, so high productivity with afucosylation technology and enhanced ADCC effector function can be offered. Besides the classical CHO platforms, we are offering, or we inherited basically the technology from Glycotope using the human K562 cell and using that for clinical manufacturing. And this is a cell line which is a niche. Whenever CHO is not functioning properly for project, we can offer the GlycoExpress technology, which is fully documented and approved by authorities around the world for clinical manufacturing. Besides that, if the customer is still not satisfied with the broad portfolio, we can offer a hack cell, and we are currently as well discussing with a multinational company to in-license a fifth platform, another human platform, to offer that for protein manufacturing. So we are expanding here in that area as well. And to come a bit then on our kind of core technology where most of the projects are running, this is, we call it chonomide. Chonomide is consisting of three different cell types. Use this one. And this is basically the key for our offering, which is a bit outstanding to other companies who are offering cell line development as well. We are offering that not only in one of these cells, but we go in parallel with all these different host cells. We are applying in this context the transposase technology, and usually for IgG-based antibodies, being that bispecifics or more complex constructs, we usually range between 8 and 12 grams per liter in titer. And in parallel, we can take a look very closely on other key issues like glycosylation. And basically, I don't know why this next slide is not appearing. This is some background on the chonomide platform. We licensed the CHO-K1 from the UK cell depository ECAC. Out of that, we generated ourselves the GS knockout. and we licensed in parallel from the couple Jason in New York, the DG44. We are having a license which allows us to sub-license to multiple tiers. So we are the only contracting partner in that context, which makes it much easier for customers kind of to get the license really as a contract in place. There's no Merck, Thermo, etc. attached. We are offering that platform together with off-the-shelf media, so very easy. Customers can go to the to the companies who are offering these media and just order it in large scale. There's no complex contracting, and in this context, we always take care of the best media combination to that clone, which is then producing the material. And via that, we really get very nice results, leading from 8 grams per liter in these clones example to over 10 grams per liter. Our current champion is currently at 12.5 grams per liter. And this comes along with 2 key factors. One is that already on the cell pool stage, we usually range between 4 and 7 grams per liter. There are outliers sometimes with 2 grams per liter, but this is kind of the mean productivity what we reach already after 6 weeks from transfection. And this pool level is a very good insight on the quality and productivity and manufacturability of the protein, which is of interest. And at that stage, the customer can decide, is the productivity high enough? Is the quality as wanted? And then they proceed to single cell cloning, and at the end, the high titer products come out. These cells then show a very high stability. So usually companies are thinking about 60 to 70 population level. In our case, we are looking even further, just to be on the safe side. It's a new system, and we did not yet see any clones which were not stable up to 19 population doubling levels. To go now into, not yet into the case study, one overview with regard to timing. We are very competitive, so very early analysis of the stable pools after 6 weeks, as mentioned. And then we have the first clones after 2 to 3 months after transfections, and the research cell bank being frozen down 4 to 5 months after starting of the project. With the stability testing, we come up to 8 to 9 months, which is as well very competitive. In that time span, you can already go into process development, downstream process development, etc. And then it's transferred to the GMP manufacturer at month 10. Now to the case study, and have a couple of minutes left. Maybe not. Annika, how many time do I have? It's 20 minutes or?
15 in total. So yeah, have 1 minute, but just show the case, and then we'll
Okay,
see how many questions.
perfect. Then I jump a bit on the final slide, which is which is here. We, as shown at the beginning, we use the Jonomide platform, and this consists of all these 3 cells. The customer basically went to intensive screening at the beginning, and then went to a GMP manufacturer who failed with their cell-based system. So they came to us, and we started very broadly to see what what is the best solution for the molecule. It was a problem in turbidity and in productivity. And then we started all 3 cells. We went into the stable pool level, and very interestingly, it could be shown that the DG44 did not work out, and that was a cell which was previously used by within the other company, because it made fragments on the light chain of that antibody. And GS knockout and K1 knockout went well, and they ranged similar in titer with roughly 6 grams per liter on the stable pool level. So we went to cloning, and we came up with the data are some slides above, that the GS knockout did not work out because it produced a lot of high-mannose. The K1 did not. So at the end, that customer was totally happy that we basically saved the project and basically saved the company, because that is a 3-product company, and that was a major product within their company. And the K1 was then currently 2 months ago transferred to the GMP manufacturer with a productivity of over 10 grams per liter. And we have already shown the stability, and the USP development was initiated. And with these guys, we are currently discussing the contract on clinical laboratory work. So to round that up, many things under one roof here at FyoniBio. And thank you for listening. Any questions?
Yeah, thank you, Lars. Are there any questions from the audience? Maybe also regarding the case study? I don't see at the moment questions. But you have shown maybe the other case study you wanted to show. Is this similar, or the very other case?
No, it's basically the same case study, but with some information on glycosylation, etc. So more.
What was the outcome there? So what challenge could you solve there?
No, that is basically just a picture what I just summarized. That when you take a look at the N-glycan profile, for example, for different clones out of that antibody producing project, we could show that the GS knockout had very high mannose structures. So just kick out these clones, take further the good clones, and they basically all came from the K1 cell line. One slide maybe further up. So all clones were quite high in productivity. So the customer could really pick then very nice product with very nice quality aspects as well, and put that to clinical manufacturing. That is maybe the full story around here.
So thank you very much.